Yesterday we were discussing about the cell division and we had a small overview of cell division. But about the cell division, I told you that I have mentioned in the previous lecture that why do cells need to divide, and what is the need of cell division, and what is the time. So okay, one I think I missed yesterday that what is the time of cell division for many cells? For example, time of cell division is actually the need of cell division, is like need of cell division. For example, if we want to replace, yes, I mentioned it yesterday, that if you want to replace, or if you want to grow the body, or if you want to increase the number of cells, then we would be performing the mitosis. So time of cell actually means the need of cell division. So I now remember that I did mention it yesterday, that why do we need the mitosis or meiosis? But mitosis is we discussed yesterday that mitosis is cell division in which the chromosome number remains the same. Uh, this is the one type of cell division. So for today's lecture, if I if I teach the mitosis completely, stage by stage, I think that today's lecture would only cover the concept of mitosis, but it doesn't matter. Let me be very clear about two concepts that are very critical in understanding the cell division. There are two words used in this topic. One is cell cycle, and the other one is cell division. Cell division. Cell division, we discussed yesterday very briefly that these are the phenomena of how the cells divide. Phenomena of how the cells divide. This is cell division, and it has two further subheadings. One is mitosis and other is meiosis. But what is a cell cycle? This is very important point. Cell cycle is the phenomena in which the phenomena in which the cells in which a cell divides a cell divides into two a cell divides into two, and next, or you also may say that, and the daughter cells divide again after some time. It is a bit ambiguous statement, so let me draw the diagram, and the diagram would explain you better about the cell cycle. Actually, suppose that this is a cell, and this cell is freshly divided. This cell is freshly divided into two. One is this, one daughter cell is this one, and one daughter cell is this one. So what would happen that one of these cells, suppose this is the one of these cells would be would start to mature. Maturation means that it is going to be the part of the body. This cell is going to be the part of the body, and this cell, this one now would again enter into the next division phase. It means that whenever our cells divide, one of the cells after mitosis, this is now mitosis, one of the cells after mitosis start to be the part of the body, or it is ready to perform the functions of the body, and the other one again enters into the division phase. So it means that both of the cells are not going to be the part of the body, or you may say that both of the functions are not going to be mature. One of them is going to be mature, and the other one is is going to start the next division, and the next division is like अब वो अगला phase start हो रहा है, ठीक है? Now it would follow some further steps, it would follow some more steps, and after some steps it would proceed to this stage again, and it would again perform the mitosis. It would divide into two, and one of them would be performing the function of the body, it would mature, and the other one would again enter into the division phase. This phenomena in which a cell divides into two cells, one performs the functions of the body and the other again enter into the cell division. This phenomena is known as cell cycle. This is a cyclic process. Cyclic process mean a thing starts from a point, performs many functions, and then end into the same point, and then again start from the same point. Now this is the origin point. This phenomena is going to be known as the cyclic process. Why? It is starting from the same point from which it finishes, and it goes again and again. There are two features of a cyclic process. One is the process that occurs again and again, and the other is the starting and ending point of the cyclic process is always same. This is the starting point as well as the ending point. This is so the cell then one of them starts to get mature, and the other one again enters into the division and performs the same activity. So you can see that it is going through a cycle. This process is known as cell cycle. What would happen? This cell would wait for this time, यहां से लेकर यहां तक, from here to here, यहां तक ये सेल जो है, this would wait for the next division. It would prepare itself, it would prepare itself for the next division, and then after that it would finally divide, performing the mitosis. So this is a very important phase in which the cell first prepares itself. You know, if the cell does not prepare, for example, if there is no preparation phase, if the cell does not prepare, what would happen? The cell would undergo mutation, and mutation is very dangerous. The mutation can occur, and mutation can lead to cancer. So in this phase, the cell prepares itself for what? For next division. For next division. Suppose I told you yesterday that if this is our DNA has to be duplicated. Our DNA has to be duplicated before division. In this preparation phase, which is starting right after the first division, and it is consuming a lot of time. If I tell you about the division of time, 75% of time is spent in preparation phase, and only 25% of time is spent in the division. What do you mean by division? Mitosis. Only 75% of time is taken in the mitosis. So it means that this phase is very really important, and this duplication occurs in the preparation phase. Duplication occurs in preparation phase. So this is a very important phase. That is why it has a very particular name. This phase has a very particular name, that is interphase. This is interphase. Interphase is a phase intermediate between two divisions. One division is this, and again the second division is this. एक सेल डिवाइड होता है और उसका डॉटर सेल दोबारा डिवाइड होता है, तो उसमें जितनी देर लगती है उसको हम कहते हैं इंटरफेस. ठीक है, तो अब आप देख लीजिए कि दिस सेल इज डिवाइडिंग, एंड देन अगेन दिस सेल वुड वेट सम टाइम, एंड इट वुड अगेन डिवाइड. सो टाइम spent between the two divisions, let me be very clear about it, time spent between the two consecutive divisions consecutive divisions is known as interface. is known as intermediate phase or interphase. Or interface. So this is what we call as an interface. And let me tell you very clearly about the cell cycle now. Let's be very specific. Cell cycle. Cell cycle is a cyclic process. Cyclic process. And it consists of two parts. Number one is interface. That is the time between two consecutive divisions. And the second phase is M phase or division phase. Division phase. M phase is can be divided into two types of cell division, mitosis and meiosis. Let me refer back to the diagram which I just drew on this page, that this is the time of interphase where the cell awaits for the next division, and the time that is taken in the division is known as M phase. So now we have all the technical terms regarding the cell cycle. Interphase, the time taken, and during the time in which the cell actually divides, that is known as M phase or mitosis or meiosis phase. So a cell cycle is consists of interphase and M phase, while the cell division consists only of mitosis and meiosis. Let me repeat this sentence. A cell cycle consists of interphase and the cell division consists of meiosis and mitosis. Let's draw a small diagram, also a small diagram here. This is the division. This is the cycle of a cell in which this cell is divided into two. Now, which location would you label as the M phase? Only this time span is M phase, in which cell is actually dividing. All this time that has been spent for the preparation, all this time it is interface. A cell undergoes the M phase, then goes into the interface, prepare itself, and then again enters into the M phase. So M phase is the division, and both of them collectively would called as cell cycle. So what is the purpose of telling all these points about the cell cycle? That suppose now that a cell is in interface. Cell is in interface, and interface के बाद क्या होता है कि the cell and going to enter into the M phase. Going to enter into the M phase. What should be the preparations? What important functions a cell has to perform before entering into the M phase? Number one, it must have duplicated its DNA. It must have duplicated the DNA. Second is, let me draw it a bit. It must have duplicated the DNA. The second is, it must have duplicated the centrioles. You know about the centrioles or not?
No.
Oh, that is a bit of problem. Okay. Haploid, mitosis. Okay. Centrioles. Centrioles are organelles of cell that create fibers to divide the chromosomes, divide the chromosomes from center. I told you yesterday that this is a centromere and these are chromatids, and this chromosome would be divided into two from the center. And as a result, this would be one chromosome and this would be another chromosome. So this division is uh, carried out by special fibers. This division is carried out by special fibers. These fibers have a specific name, and I will tell you later. But these are the fibers that are made by the centrioles. And centrioles are very specific organelles. Organelles are like small organs. You know the definition of organelles, right? So these are okay, you didn't answer. You know the definition of centrioles? Oh, sorry,
Centrioles, yeah.
organelles, organelles.
Yes, they're the parts
Okay,
of a cell.
parts of the cell. So centriole is another part of a cell, and it creates the fibers that would be joining with the centromere or central point of the chromosome, and then it would pull the both the chromatids apart, and it would separate the chromosomes. So what I mentioned here, before entering into the M phase, the preparation should be the DNA should be duplicated and the centrioles should also be duplicated to perform the proper functions. So first step of now the cell has entered into the M phase. First step is first stage is prophase. During prophase, some of the very important phenomena appear under the microscope. Okay, let me tell you one more thing, one practical aspect of all this phenomena, that we are observing all this under the microscope. This diagram has been taken from the from the observation under the microscope, and this actually appears like this. It actually appears like this. If you create a proper slide, for example, if you take a dividing cell, there are multiple dividing cells in the plants. So if you take the dividing cells from the plant, put those dividing cells under the microscope and examine them, you will come to know that these would, while performing mitosis, this type of specimen would appear under the lenses of microscope if the cell is going through the mitosis. Only and only if the cell is going through the mitosis, you can see the cells like this. Okay, let me make it a bit more clear. During the prophase, number one, the nuclear membrane disappears. Nuclear membrane disappears. Why the nuclear membrane has to disappear? Let me make it clear. This is a cell. Before entering into the M phase, the cell enlarges. It has to enlarge. So it can be easily divided. So once the cell has enlarged, the the nuclear membrane has to disappear. This is the nuclear membrane. NM, I'm writing it. And the chromosomes are inside. The chromosomes are inside. You can guess that why do you have to remove the nuclear membrane? Can you guess?
So that the chromosomes can divide.
So that the chromosomes are attached with the fibers, with these fibers, and then these can be pulled apart. So this is why the nuclear membrane has to disappear. So nuclear membrane disappears in during the prophase. What does it look like? Let me make a sketch of that as well. So this is the cell, and after disappearing of the nuclear membrane, the chromosomes spread in the cytoplasm like this. Chromosomes spread in the cytoplasm. The centrioles present at this end. Centrioles come at this end, and they now it's their turn. They will throw their fibers towards the centromere of the chromosome, and they will align them. Let me show it. How will it happen? So these are the fibers of the centrioles. They have thrown it towards the chromosomes like this. This would be throwing it from this end and attaching it on this end. So you are well aware that there are two chromatids. So one fiber should be coming from this side, and the other fiber should be coming from this side to pull those both the chromatids apart. So the centrioles from the both ends would throw their fibers towards the chromosomes, and these fibers would attach to the centromere point. And now what would happen? This is very interesting point, in fact, that the next phase is metaphase. During the metaphase, there is a phenomena known as alignment. Alignment. Now, what is alignment? That all those scattered chromosomes would be aligned on the central axis of the cell. These all would be aligned in the central axis of the cell. Now you can easily observe that what is the difference between the cell of a prophase? This is prophase, right? The scattered chromosomes are here, but in the during the metaphase, all the chromosomes would be aligned, and these would be aligned by these fibers. First, these have to be aligned. So these fibers would cause the alignment of the chromosome at the center, and this is the metaphase. Now, this is the alignment, and then in the next phase, anaphase, which is anaphase, the separation occurs. Separation occurs, and how the separation would occur? Let me draw it. This is a cell, and these are the centrioles. These are the fibers of the centrioles, and these are the chromosomes. These have been pulled apart. Now these all have been separated from each other. After the alignment, now these have been separated. And then the last phase comes, which is telophase. Telophase is the phase in which all the chromosomes, now be very focused, all the chromosomes have reached the opposite poles, first, and then the nuclear membrane reappears. Nuclear membrane reappears. Reappears. Now let me draw the cell of telophase. This is the cell of the telophase, and the nuclear membrane is here. This is one nucleus, and this is other nucleus. The chromosomes have reached to the opposite poles. This is one pole, and this is the other pole. Now the chromosomes are here. Chromosomes are at home, like this. And now a step would occur that is known as a very important step, that is known as cytokinesis. Cytokinesis. Cytokinesis is uh, division of cytoplasm. Division of cytoplasm means The cell would be divided from the middle. Cell would be divided from the middle, and after the completion of cytokinesis, cell would be looking like this. One is this and the other one is this. Both have their separate nuclei, and all of them, both of them would have 46 chromosomes with them. Why these would have 46? Because in during this alignment, on the central axis of the cell there are 46 chromosomes. So when there are on the central axis there are 46 chromosomes, when these will be pulled apart, then uh, this is going to be 46 divided by 2, but 46 into 2 divided by 2. These are 46 duplicated chromosomes. So once these are pulled apart, all of these would be like this: 46 single here, 46 single chromosomes here. What which we mentioned in the yesterday's lecture, that the chromosomes first these would be duplicated, and then these would be divided into half. So from the center of the all chromosomes, now the fibers has pulled all the chromosomes from the center, and 46 chromosomes are on the left side and 46 chromosomes are on the right side. So it means due to alignment of 46 duplicated on the axis. These are 46 duplicated on axis. So while these are 46 duplicated on the axis, what would happen? Now these would be converted into single form from the duplicated. So 46 would be here and 46 would be on this pole. And at the end, both of these would have the 46 chromosome as equal as parents. Clear?
Yes.
Okay, good. Acha ji. Next. Now this is mitosis. What is meiosis? There is another thing I wanted to tell you, that I told about the divisions of the phases, M phase, sorry, cell cycle. Then I told about interphase and there is M phase. You can easily observe that M phase has further divisions. M phase has further divisions. Let me tell you about this. If I divide the M phase into further divisions, then one of M phase is divided into mitosis or meiosis, right? I have mentioned this earlier, that M phase can
Yes.
be mitosis or it can be meiosis. Mitosis is also divided into prophase. Okay, let me be more specific. Mitosis is divided into two phases. One is karyokinesis. I have not told you about this, and the other one is cytokinesis. Karyokinesis is when the nucleus divides. Karyon means nucleus and cyto means cell. When the nucleus divides into two, coming back to the diagram, you can see there is a single nucleus. And after completion of telophase, there are two nuclei in a single cell. So it looks like this nucleus divided into two nuclei after the telophase. After the telophase. So this is the completion of karyokinesis. It means that the nucleus has now divided at this stage. So one step is karyokinesis, and karyokinesis is further divided into four phases. One is prophase, then metaphase, then anaphase, and then telophase. And after completion of telophase, the cytokinesis comes, in which the cell is divided into two further cells. Once the nucleus has divided, then the cytokinesis causes the cell to divide from the middle, and the cell completely divides along with its nuclei. So this is these are two phases in which phases of mitosis in which one is karyokinesis, nucleus divides, and the other one is cytokinesis. Same happens for the meiosis, but for meiosis we have to think a bit different. Meiosis is divided into two further divisions, meiosis 1 and meiosis 2. In meiosis 1, it has separate karyokinesis and then cytokinesis. And meiosis 2 has its separate karyokinesis and cytokinesis. So, to differentiate between the karyokinesis of meiosis 1 and 2, you will have to give them the number as same as this meiosis 1. This is karyokinesis 1, this is cytokinesis 1. This is karyokinesis 2, this is cytokinesis 2. And and their further divisions would also be would also be labeled with this number. For example, the prophase of karyokinesis 1 is prophase 1. The metaphase of karyokinesis 1 is metaphase 1, same for the anaphase 1 and same for the telophase 1. So this is, and then this would be prophase 2, metaphase 2, anaphase 2, telophase 2, same for the all of the subphases. But why does this happen? Why the meiosis is divided into meiosis 1 and meiosis 2 while the mitosis does not? You remember from the previous lecture? Let me show you the previous lecture. Let me show you the previous lecture. In previous lecture, we said that the cell divides into 23 and then again into 23. We mentioned it. This
Yes.
it is like, so this is meiosis 1 in which the cell has to divide into 23 duplicated forms first, and then after that it would be performing the meiosis 2 in which the duplicated chromosomes would be again divided into the single form. So it does not happen in the mitosis. In mitosis, the chromosomes simply come into the middle, sorry, simply reach on the axis and divide it into two. But in meiosis, it does not happen. Why and how it does not happen? Let's make it clear. Let's start the prophase one, because we are not meant to study the interphase in detail because interphase is very important phase, but uh, we can mention it here that interphase has occurred. So let's go to the karyokinesis. Karyokinesis 1. In karyokinesis 1, we have prophase 1 here. During the prophase 1 of the meiosis, a very important phenomena which I did discuss yesterday, it is it occurs. How? First of all, in the prophase 1, the nucleus grows in size. The nucleus grows in size. In the prophase of mitosis, the nuclear membrane disappeared suddenly, but not in this case. In this prophase, what would happen? That let me let me clear one more thing that I have told you earlier, that if we have, this is chromosome number 1 of first 23, then this is the chromosome number 1 of the other 23. These are homologs, and these both would be going through the duplication phase. So it means that both homologs would be multiplying into multiplying, and these both would be seen as duplicated chromosome. Now these both are homologs. This is one homolog, duplicated homolog. This is other duplicated homolog. So what would happen in prophase 1? The homologs, the homologs perform the crossing over. The homologs would perform the crossing over. How? The homologs would come to each other and these would be joining their chromatids. We discussed this yesterday, right?
Yes.
After at the end of the at the end of the prophase 1, at the end of the prophase 1, the chromosomes would look like this. This is the start and this is the end. The chromosomes would look like this. The nucleus is still here. This is a big difference between the prophase of mitosis and the prophase of meiosis. That nucleus is still intact, but the chromosomes are in this form. One, they have. Oh, sorry. They have crossed their segments. This would take time like yesterday. It has taken a segment of black, and the other one that is still attached to it. Focus on the diagram now. I'm going to zoom it after drawing. They have exchanged their segments and they are trying to push themselves away from the each other, but still joined at this point. Still joined at this point. So it means that the chromosomes have done the crossing over in the prophase 1 and now they are joined at this point right now. And in this condition, when they are still joined with each other, in this condition, the metaphase starts. In metaphase, two important events occur. One is nuclear membrane. Here, it would disappear here. And in mitosis, it disappeared in prophase. But this time, it would be disappearing during the metaphase. And another important thing would happen that these would be aligned. Now, how would these be aligned? Let me draw the diagram here. This is a cell. And nuclear membrane has, oh, sorry, very bad handling. This is the cell. And now the situation of cell is like this. Let me draw it straightforward. You know that they have done the crossing over. So it gets boring every time if we differentiate between the segments. They have done their crossing over. So now these are looking like this. And the centrioles have aligned them on the center. Centrioles have aligned them on the center of the cell. Now, this is the most critical point. This is the most critical point of meiosis. Why? The arrangement is like this that 23 are arranged on the left and 23 duplicated are arranged on the right. Did the same happen in the mitosis? No. In the mitosis, all of the 46 were at the middle of the cell. Look at this diagram. All the 46 are in the middle of the cell and these would be pulled apart. But uh, contrary to the meiosis, in which 23, due to crossing over, this occurred due to crossing over. They faced each other, they crossed their segment. None of such thing happened during the mitosis. But due to crossing over, they have to face each other, and while facing each other, 23 homologs arranged on left side and 23 homologs arranged on the right side. So 23 are here and 23 are here, and now you can guess if they are separated in anaphase, what would happen? Can you tell me?
The chromosomes would separate.
But what is unique about the separation of chromosome this time?
They're having a part from the other chromosome.
Okay, one thing. The other important point is they are in the duplicated form. After being pulled apart, they are in the
Yes.
duplicated form. Going back to the mitosis, you saw that after pulling the chromosomes apart from each other, all the chromosomes were in the singular form. In this case, all the chromosomes are in duplicated form. And when the at the end of the metaphase, now this is very interesting. When the metaphase occurs, same metaphase occurs like same as like the mitosis, that the nuclear membrane would appear, nuclear membrane would reappear. Chromosomes are here, but chromosomes are different now. Why? In the metaphase of mitosis, all the chromosomes that reached the opposite poles were in singular form. And another thing, tell me, how many chromosomes would be coming to this pole and how many would be coming to this pole?
23 to that pole and 23 to that pole.
Contrary to mitosis, what happened during mitosis? How many arrived at both poles?
46.
Yes, 46 arrived, but right now 23 duplicated arrived at this pole and 23 at this pole. Now you can see that both of these nuclei are containing 23, but in duplicated form. But in duplicated form. Now, now, let me tell you one thing. Let me tell you one thing. After that, what would happen? The cytokinesis would occur. The cell, both of the cells would be divided into two cells here, but these are now 23. Same as this one. Same as this one. Okay. Now, now this would there would be another interesting thing. This would perform another meiosis, and that is meiosis 2. Now this is meiosis 2, and during meiosis 2, now you can guess what would happen. It would be as same as mitosis. Why it would be as same as mitosis? Because if you examine the metaphase 2, these are prophase 1, metaphase 1, and then anaphase 1, and then oh, so this is not metaphase, this is telophase. Why am I writing the metaphase? Metaphase 1. Okay, metaphase 2, during the metaphase 2, you can see that there are chromosomes aligned on the center of the cell as same as the alignment during the mitosis, as same as alignment during the mitosis. And but now how many chromosomes are there in the center?
Um,
Metaphase 2, continuation of meiosis 1. So it is meiosis 2. Meiosis 2 is okay. Okay, let me make it easier for you. Meiosis 2 is occurring both in these cells. Meiosis 2, meiosis 2. So now, how much chromosomes would you think would be at the center of the cell?
23.
23. So it means if the fibers come and join at the chromosomes at the center, what would happen? These would pull the chromosome as same as mitosis, but the chromosomes are not 46 at the center. These are 23 because uh, these cells are performing meiosis 2. If these cells are performing meiosis 2, it means that these have 23 duplicated and all those 23 would align at the center, and after the separation, all of these would get single. And when these get single, These would be again 23, but in single form. This would be again 23, but in single form. So it is like this. We did discuss it yesterday. These would be separated, and all the gametes would be having the 23 chromosomes in the singular form. And finally, it would be turning into the gametes. Okay. They have shown crossing over here actually. Showing crossing over here. Now this would cause, I told you that yesterday that the crossing over would cause the variation. Why? Because the chromosomes have exchanged their segments. It would cause the variation. Clear?
Yes, sir.
Okay, good. It is good that we have discussed the mitosis because for further topics, especially for mutation and for evolution, the meiosis and the variation in meiosis is very important. So it is good. Anything else?
No, sir.
Okay, great. So, okay, beta, Allah Hafiz.